la première chose que je veux dire, c'est que je remercie Dr. Farge pour l'invitation et que c'est vraiment un plaisir pour moi d'être ici et de parler de ce qu'on fait dans les labos, mais aussi d'apprendre de vous. Et je m'excuse, mais je vais changer maintenant l'anglais parce que je me sens un peu plus à l'aise avec l'anglais. Pas beaucoup, parce que mon première langue, c'est l'espagnol, mais on va faire l'attente. Alors, I don't have any disclosures, and this is what I would like to do within the next 15 minutes. The first thing is that I would like you to switch your brains, because we are not longer talking about hematopoietic stem cells. We are going to start talking about mesenchymal stromal cells. And I'm going to start by defining them. I'm going to show you a little bit about what we do when we talk about licensing or priming of these cells. I'm going to give you some very basic concepts about extracellular vesicles. And the question that we are working on essentially is what is the effect of cytokine priming on MSCs with regards to vesiculation. And I'm going to refer this to the beautiful relationship that MSCs have with T cells, which in general, MSCs are able to inhibit the proliferation of activated T cells. And because of that, we think they are useful in different settings. So I apologize for those of you that this seems obvious, but I really think that it's important that we keep in mind that mesenchymal stromal cells, at least a big portion of them, are pericytes. And what that means is that our cells that are surrounding small caliber vessels, and they're able to promote their stability. The images that you see on your right are images from a fantastic paper from the WIT that essentially show that MSCs can be seen in every single organ, not only of adults, but also from fetal embryonic and from embryonic tissues. What differs is the way these MSCs are distributed and also the amount of MSCs that you find in different tissues. When we get MSCs in the lab, we characterize them based on criteria, which are their capacity to adhere to plastic, their capacity to differentiate into at least 3 lineages that depends on what we provide to these cells in vitro, and their surface phenotype that essentially should not be expressing markers that are expected to be seen in hematopoietic lineages, and they do express some markers that are also seen in cells from fibroblastic lineages. And this is key, because when we in the lab are using MSCs, we are dealing with a group of cells that are highly heterogeneous. So there's, at this point, not a single marker that specifically can tell us that we are working with a better quality product or not. And that's something that really we're trying to get to that. What we know is, despite of their heterogeneity, that MSCs do have different functions. And because of this, they have been used for different types of clinical applications. The 3 most important functions are their capacity to induce tolerance or immune tolerance, their capacity to help with wound healing. And this is a very interesting thing, because on one hand, they promote wound healing, and on the other hand, they do have antifibrotic effects. And the third thing is their pro-angiogenic capacity. Probably the best characterized function in terms of mechanisms is their tolerogenic properties. And that's why I'm referring specifically to that today. And what we have learned during the last 10 years is that the functional effects of MSCs are not related to the engraftment of these cells or to the interaction between cell and cell directly, but they are related to the capacity of MSCs to produce certain factors in their secretome, and those are both soluble factors and extracellular vesicles. Why do we know that soluble factors are critical for the function of MSCs? This is one of many studies that have shown that the effect size of MSCs is similar to that of using conditioned media, and this specifically refers to lung diseases, but again, this has been shown in different other models. one of the key things that we need to understand when we think about the function of MSCs is that these cells are not constitutively immunosuppressant. So MSCs are going to acquire this function when they are in an environment that is stimulating their activation. How do we do this in vitro? Essentially, we do use interferon gamma and TNF. And how do we know that when we expose an MSC to these cytokines, that cell becomes activated? Simply by looking at their surface expression of HLA-DR. So an MSC that is resting does not express HLA-DR. An MSC that has been primed by these two factors, they do have a significant increase in that surface marker. So what happens after an MSC is primed? So what they change is the composition of their secretome, the soluble factors that they produce, and essentially that leads to an increase in the amount of soluble factors that have to do with an anti-inflammatory effect, such as, for example, IL-10, PGE2, IDO. And how do we know that that happens is because when we look at what is the capacity of the secretome of MSCs to suppress activated T cells and the resting conditions, there is some effect, but clearly that effect increases dramatically following the priming of MSCs. So I said that MSCs exert their functions when they become activated, and that is mediated by changes in their secretome. I showed you what happens with the soluble factors, but we did not know much about what happens with MSCs post-activation in terms of extracellular vesicles. So what I'm going to do in the next few minutes, or with the next few slides, is to show you what an extracellular vesicle is, why a cell cares to produce extracellular vesicles, and at the end I'm going to show you the so what. Why is this important for us to know? So extracellular vesicles are particles that are normally produced by every single cell, that they are delimited by a double layer of lipids, and they cannot replicate because essentially they do not have an intact and functional nucleus. In general, extracellular vesicles have been characterized depending on their diameter. And based on that, vesicles that are more than 200 nanometers were called microvesicles, those that were between 50 and 200 exosomes, and those that were more than 1,000 apoptotic bodies. However, we know currently that this definition, especially to differentiate microvesicles and exosomes, is not optimal. And the reason for that is because you do have several microvesicles that are in the 200 nanometer range, and you cannot say that if you have a group of vesicles that are in that range, they are definitely exosomes. So why is this important, and how do we really can differentiate microvesicles and exosomes? Well, the answer is based on their mechanism of production. So microvesicles come from the budding of the plasma membrane, and then the liberation by shedding of those vesicles, where exosomes is exactly the opposite, is the invagination of the plasma membrane with formation through the endocytic pathway of an early endosome, a multivesicular body, and this is what either goes to fuse with lysosomes and goes to be degraded, or whether that is eliminated from the cell from the process of exocytosis with the liberation of exosomes. I am not going to refer to apoptotic bodies now, but there's more and more evidence that apoptotic bodies are not always this size, and even there are vesicles of the size of exosomes that are liberated following apoptosis. So why a cell produces extracellular vesicles, and this is exactly the same reason why you communicate with each other. Extracellular vesicle production is a key mechanism of cell-to-cell communication, and that can be altered after specific signals. One of the things that I think it's really important to consider is that if this is a schematic representation of an extracellular vesicles, what these vesicles can communicate between one cell and the other one are specific proteins that are either in the membrane or inside the vesicle. They can also communicate through certain RNAs, either microRNAs or non-coding RNAs. And there's also, in certain cases, specific nucleic acids that are exchanged as well as lipids. So how does this vesicle get to modulate the function of a different cell? So what we know currently that there are at least 5 or 6 different mechanisms by which these vesicles can enter a cell, either by diffusing through the lipid membrane, either by active processes such as endocytosis that could depend or not on caveolin. And there are other mechanisms that are currently under studies, but essentially they can also act directly on the surface of the cell through the receptors for specific proteins in the extracellular vesicle. So let's go back to the question that we asked in the beginning, which is what is the effect or the impact of MSC activation on the production of these cells? So if you're studying that in your lab, the first thing that you need to do is be able to isolate extracellular vesicles from your MSCs. I showed you how you characterize your MSCs, and what you do is you essentially put a specific number of MSCs to condition media, and then you get that. There are different ways in which you can generate extracellular vesicles. We use differential ultracentrifugation, which is a well-accepted method to do this. Once you isolated your vesicles, one of the first things that you want to know is what is the size of the vesicles that I do have. And you do that with something which is called nanotracking analysis, which allows you to see essentially what is the number of particles that you have in your suspension and what is the size of those particles. As you can see here, after we they did ultracentrifugation of the conditioned media by MSCs, we see that most of the particles that we're able to isolate were in the 200 nanometer range. The second thing that you do is you divide these extracellular vesicles in a gradient of sucrose and their specific density that is expected for small vesicles. The third thing that you do is you look with electron microscopy at the structure of those vesicles, and essentially, I'm not sure if you're able to see it from where you are, but what you see here is essentially that double layer of these small particles. And finally, you look at specific proteins that are present in the external aspect of extracellular vesicles, such as CD81 and ALIX, and you really do not want to see in your extracellular vesicle component any proteins that are present in other organelles of the cells, such as in the Golgi, the presence of calnexin. One of the things that it was really interesting is that we do have currently the capacity to look at very, very small particles. Imagine this is much smaller than a cell by a specific type of flow cytometry. And here what we can confirm is that the vesicles that we isolated express proteins that are expected to see in exosomes such as CD83 and CD81. And they do also express proteins that are expected to be present in MSCs, such as CD90 and CD73. So clearly, we can confirm based on that the extracellular vesicles have an origin from MSCs and that they do fulfill all the criteria that are required currently for calling an extracellular vesicle as such. So once you're certain that you have what you want to study, then you give the stimuli that you're interested in learning about. And this is what we have done. So we exposed the EVs to either interferon and TNF, which is this curve, or we look at them under resting conditions. And what we've learned from this is essentially by doing NTA that the amount of vesicles that are produced following activation of MSCs is much higher than under resting conditions. However, the distribution of the size of the vesicles is similar. This is essentially a summary graph of this data, and there are 6 samples included here. So you know that activation of MSCs leads to an increased production of extracellular vesicles, and the question is, they are small extracellular vesicles, but by which pathway these vesicles are produced. I put this small because I don't intend to give all the details about how an extracellular vesicle is secreted, and this is the endosomal pathway, but I want you to trust me that there are key proteins that are needed for a vesicle to be expelled as an exosome. And those are tetraspanins, those are proteins that are called escort proteins, and those are the ones that we have specifically looked at in this custom array. And the question that we asked here was, is there any difference in any of these proteins between samples that are eliminated after activating MSCs? What we found is that, well, one of the differences is that there's an increase in CD82 in the vesicles after priming. And again, this is just a reflection of the fact that there's more extracellular vesicles post-priming. And the other thing that we have found is that there is an increase in a protein which is called RAP27B. Why this is important? Because RAP27B is a fundamental protein that is associated with the fusion of the multivesicular body to the membrane in order to liberate exosomes. So because of that, because we proved that these small extracellular vesicles are regulated by the endosomal pathway, we can call them exosomes. The next thing that we wanted to look at is, besides the increase in the number of extracellular vesicles that are produced after activation of MSCs, is there any difference in their cargo? So essentially, are the proteins within the exosome different when they are generating from a MSC that is under resting or in the context of inflammation? So we found that there were at least over 180 proteins that were uniquely expressed in extracellular vesicles from primed MSCs. And we looked at the different biological processes in which these proteins were associated. And particularly, we were interested in those proteins that had to do with inflammatory responses. Among them, we were very surprised by the fact that there were two well-known proteins that are related to inflammatory processes. One is called A20, and the other one is called TSG6, TNF-stimulated gene 6 protein that were increased in extracellular vesicles from primed MSCs. We confirmed that by Western blot, and again, this is extracellular vesicles from resting MSCs, and this is from activated MSCs, and as you can see, there's a clear increase in TSG6 protein, and the same thing with A20. And essentially what we confirm with this is that the content of the vesicles changes following MSC priming, and there is an enrichment in exosomes of proteins that are critical for the modulation of immune processes by MSCs. Of course, the question that you can ask yourselves is, okay, that's fine. There is something that changes in these extracellular vesicles, but it's that important to function. So in order to look at that, we tested the capacity of exosomes from primed MSCs to suppress activated CD4s. And we showed that there is a dose-dependent increase in CD4 suppression when we used only extracellular vesicles from primed MSCs, and that was not dependent on a difference in MSC viability. So clearly what we show is that the extracellular vesicles produced from primed MSCs are clearly by themselves able to inhibit T-cell proliferation in a dose-dependent way. The last experiment that we wanted to prove was the relationship between the RAP27B increase and the different cargo in the MSCs. So what we did is we used a way in which we inhibit RAP27B in MSCs. This is called small interference RNA. And we looked at the way those modulated MSCs conditioned the media. The first thing that we found is that there was a reduction in the number of extracellular vesicles produced by samples in which RAP27B was inhibited, and that was not something that was unexpected. And this is what you see here in terms of a summary graph. But the interesting thing that we also confirmed is that these vesicles that were produced in the context of SI RAP27B were reduced in TSG6. So again, what we can see, or what we've learned here, essentially is that following cytokine priming, RAP27B upregulation promotes the secretion of TSG6-loaded extracellular vesicles. So what I've shown was that the immunomodulatory properties of MSCs are not constitutive, and that they are induced by the inflammatory microenvironment, and we call that priming or activation of the MSC. That priming shapes the MSC secretome, not only in terms of their composition of soluble factors, but also extracellular vesicles. That MSC priming promotes RAB27B dependent vesiculation, and because of that, we can say that the increase in extracellular vesicles is particularly an increase in exosomes, and that it enhances the anti-inflammatory cargo of these vesicles with the increase in TSG6 and A20. And finally, that EVs from primed MSCs are able to inhibit T-cell proliferation, which confirms the idea that subcellular products are effectors of MSC function. The question that always I ask myself and my students ask me is, so what? And I really believe that this is the answer in this case to that question. I really believe that as long as we do not understand how cells or cell particles function, we are not be able to translate these into clinical trials that are really effective. So I think that that is the value and the importance of doing basic research to support the clinical trials that later on can be performed. This is the most important slide that I have. I really want to thank Anastasia Cheng. She's a master's student in my lab. She finished her master's, and all the data that I presented was really produced by her. So this is for Charlotte specifically. This is how much we can learn with a student within two years. I am very lucky because I work in the same institution than one of the leaders in the field of extracellular vesicles, which is Janusz Rak, and we do have a very good collaboration together. And these are the institutes that are funding my work. This is the way I met Dominique Farge even before the time that I really met her, which was in 2016. And if I have to say, what did I learn from Dominique? I have to say that probably she's my best example of how to stay strong, stand up, and have a voice. Thank you so much.